start. So this time I won't go into the PowerPoint mode so that my slides will be visible. So whenever any problem happens, then you can write in the chat. I'll see now. Okay, so in today's session we'll look into the week 4 topics. So in week 4 you have looked into the mass spectrometry hands-on. You have checked that how to analyze different spectra and means how to look at the peaks and how to infer the peptide sequences by looking at those peaks. And then you have also looked into the challenges which are faced due to the measurements when they are taken, what kind of liquid chromatography gradients are used. So on the basis of there could be differences among different batches or different samples. So to make the comparison at the normal ground, we have to normalize the data so that we can make the better analysis and better comparisons. So normalization techniques you have seen batch correction, that if different batches are collected at different times, so you have to make sure that whether it is a batch effect or are the components or the proteins which are present, they are showing the differential effect. So that thing we have to go through. Then missing values, so whenever you take readings there could be some missing values and there could be multiple factors for that, so you also have to deal with these missing values. And you have also looked into different statistical tests for means hypothesis testing, and when you are comparing different samples and you are saying that there is a difference between both the samples, for example comparing normal person with the cancer patient and saying that these particular proteins act as a biomarker, so in that cases we need to perform statistical test so that we can confidently say that our hypothesis is correct. So all these things have been covered in week 4, and in today's session we will be practicing few MCQs along with looking at the current literature, and I'll take your doubts. So firstly we'll go through the evolution of mass spectrometry, that how it has evolved through time. So firstly the first concept of this mass spectrometry, so first concept was given by Wilhelm Wien, who said that in the presence of magnetic field the charged particles they get deflected. So this was the first idea, that we could use the magnetic field to see the deflection, because each electric particle will be deflected by some different rate. So in that way we can distinguish between different particles or different ions, you can say. So the concept was introduced by him, and then J.J. Thomson came up, who has given the concept of electron, and he also was the first one to give the first initial mass spectrograph idea. And then he said that the particles could be distinguished on the basis of their mass to charge ratios. Then came Francis Aston, who is known as the first person who who discovered the first mass spectrograph, because J.J. Thomson discovered it, but that was the initial step, that was not a complete mass spectrograph, but Aston was the one who designed the complete mass spectrograph. And later on, after Aston, different advancements happened, different techniques came up, and then some corporations such as Consolidated Engineering Corporation, they were the one to market that mass spectrograph so that more and more people could use it. So at that time it was quite advanced for that time, in 1940s, so it was advanced technique. Then later on, slowly and slowly, different mass analyzers came up, such as time of flight analyzer came up, then quadrupole came up, then ion trap came up, then later on MALDI came up. So in that way advancement happened in the field of mass spectrometry, so that the proteomics can be studied with better resolution, which was not previously possible with the help of those antibody based assays. So mass spectrometry was a better measure, so that we can look at the different peptides and different proteins, we can directly sequence them with high confidence. So this was how the mass spectrometry evolved. And then so if we look at the origins, it originated in 20th century, the concept. So it was the concept was introduced by J.J. Thomson and Francis Aston, who were they were the pioneers in this field. Thomson discovered the electron, and he said he was the first one to design the crude mass spectrometer, and which used to measure the mass to charge ratios of ions. Later on, Aston was the one who discovered the isotopes, because isotope concept is also important in mass spectrometry. So isotope concept was introduced by Aston, and he also earned Nobel Prize for the concept of isotopes, and he was the one who discovered the complete mass spectrometer, which could separate different isotopes of the same element. So after the discovery of this Aston mass spectrograph by Aston, the mass spectrometry picked up pace and this field advanced. So if I have to mention in small or in one line about the mass spectrometer, so the basic concept of this mass spectrometer is just to generate multiple ions and then to compare or to separate them on the basis of their mass to charge, so that we can capture the relative abundance of each ion. We can infer the sequence of atoms or sequence of amino acids or residues that are present in a particular peptide if we talk about the proteins. So all those things can be done with the help of mass spectrometry. Okay, so this is the concept picture of how mass spectrometry happens, how the data is analyzed in that. So firstly, we have a biological sample which could be a case versus control, because if we have to compare between two samples, so we will be having a case and we will be having a control. So that could contain the protein mixtures which could either be obtained from biofluids or tissue lysates. And then first step is to digest them into peptides, because we cannot analyze entire protein, so we have to break it into peptides so that they could be further ionized into smaller ions. And so first step is to digest the peptides or to fractionate these peptides. If they are still long enough, then we have to further fractionate them. Then we pass that sample through liquid chromatography, where the separation happens. So it depends on what kind of liquid chromatography you are using. So for example, in reverse phase liquid chromatography, the separation will happen on the basis of the hydrophobicity of those peptides. So higher the hydrophobic it is, higher, so slower it will move through that chromatographic plate, and otherwise if they are hydrophilic, then they'll elute faster. So on the basis of that, the separation will happen, and the next step is to perform mass spectrometry. So after the separation has happened, you will allow the sample to pass through mass spectrometer, and what it will do, it will further ionize that. Those ions will further pass through the mass analyzers, which will analyze the mass to charge ratios, and ultimately detector will capture that what are the ions, it will detect it. And then I also want to clarify that whenever you see MS together, it means that it is a tandem mass spectrometry. So tandem means whenever multiple mass spectrometry goes step by step. So it's not just a single mass spectrometer, means not a single ionization step is followed, but you are going through multiple ionization stages. So in triple quadrupole, you go through the ionization step 3 times. So firstly you will ionize the sample, then later on it will separate, mass analyzer will detect, and ultimately you are again passing it through another ionization chamber, where it will further fragment down. So in that way it improves the resolution, it will precisely give you the information, because sometimes what happens, if you have very large protein and you are passing those larger proteins, you have some peptides, you are passing them through first ionization chamber. So in that way you will not be able to compare properly, because the ions will be large. So in that case the fragmentations are not that well to study. So what happen, we use another mass analyzer so that we can further fragment them. So at each step we perform some kind of selection, we'll select some mass to charge ratios. So we'll see that all these peptides have same mass to charge ratio. Okay, let's pass them through second mass analyzer. So in the second step they will further ionize it into smaller fragments, so that even if we have peptides of similar mass to charge ratios, but still in the second round we'll be able to detect properly. They could be different peptide sequences. So in that way this tandem mass spectrometry is helpful to increase the resolution, and distinguishing ability also increases with this. And ultimately we perform data analysis, in which after mass spectrometry we have obtained the peaks on the basis of the mass to charge ratios. So in data analysis we have to label those peaks, and we have to identify that what could be the peptide sequence, what could be the amino acids present in the peptides. So these all things are done in mass spectrometry, and how the data is analyzed, and it gives us the peptide identity. It tells us that what is the sequence of peptide. If you have the sequence of peptide, then you can get the sequence of protein. And as we have discussed in the previous session also, that this mass spectrometry, it takes the help of already available database. So we already have human proteome database, in which we have the sequence of entire human proteome. So whenever you obtain the peptide sequence after the analysis, you will compare it with the database, and you will see that whether that sequence is present in the database or not. So if it is present, then only we will be confident enough that the mass spectra has generated the right spectra. So in that way we compare it, we will see that whether our spectra is means relevant or not relevant. So with that we can also identify that which protein is corresponding to that peptide. So in that way we will be able to identify the protein as well. And we will also compare the relative abundance, because if we are performing labeling or we are using certain label free methods, then also we can compare that certain peptides could be expressed in greater quantities, which can give us the information that maybe this protein is more abundant in the sample because its peptides are more abundant, its peptides are more enriched, the peaks are more, means you can compare that. So if greater concentration is present, so that thing you can also use it for the relative abundance comparisons between different samples. So now the basic thing which we have looked in mass spectrometry is the fragmentation, because fragmentation is very important step if we talk about mass spectrometry. So what is fragmentation? So fragmentation is that when the peptides, they are broken into smaller pieces, they are generally broken into ions, because peptides are the samples which you are providing, and then those peptides are further broken into ions, and those ions are known as fragment ions. So these fragment ions will correspond to the, means it will be a chain of different residues or different amino acids, it is not a single amino acid. Okay, so that's why these are the fragment ions, because it has a fragment of a peptide which is not a single amino acid, it will be a fragment ion, and this is used during the mass spectrometry. So the pattern of fragmentation, it provides you critical information about the peptide sequence and structure. For example, in this figure if you see, so this is my peptide sequence which is DEFGH. I am performing its fragmentation. So what fragmentation will do, it will break down it into a smaller fragment ions. And this fragmentation also, in the previous lecture we have seen that how different ions are produced. So there are different methods through which this electrons pre-ionization or other methods through which these bonds are broken, and after breaking the bonds we are able to see the fragment ions. So first thing is that in the first ionization, so first fragment which could be formed is like DEFG, and then H will be separated, or either it could be DEF and then GH are separated, or H or G are separated, or D is separated. So in that way different fragments could be generated in mass spectrometer, and after that mass spectrometry will look for the peaks. So there will be different intensities of each peptide, because the peaks will be the mass to charge ratios. So mass will be higher if more and more amino acid or more and more residues are present, so mass will be higher and charge will be comparatively less. So in that way you can distinguish, because the last, if you see here in this right hand side, the peptide which corresponds to the complete entire peptide which was used in the experiment, so it will have the highest mass to charge ratio. So this mass to charge ratio will give you the idea of the length of the peptide, or it would help you to identify or distinguish between different peptides. So in that way, by comparing all these peptide sequences and their mass to charge ratios, by taking the difference among consecutive mass peaks, we can identify that what could be the amino acid which is present. So the key points that why we use fragmentation, so it helps us to determine the sequence, and the specific masses of fragment ions correspond to the sequence of amino acids in the peptide, which help the researchers to deduce the protein or the peptide sequence. And it can also help us to localize the modifications, that where the modifications are present, so that also we can pinpoint by looking at the fragments which are generated, because their mass to charge ratio will vary. Because in the last presentation also we have seen that if there is some post translational modification which could be phosphorylation, acetylation, so in that way if we fragment it, then its mass to charge ratio will change, its mass will increase. So in that way we'll be able to distinguish, because we already have a table which contains the information of all the theoretic masses. So in that it tells us that this could be the theoretical mass, and if its mass is higher, then we can kind of infer that what could be the functional group which is present in that peptide. So in that way we can look for the post translational modifications, and even we can also pinpoint the locations, because when you will be looking at different peptide fragments, you will be able to know that which position of the peptide contains that post translational modification, because other fragments will also be present. So comparative analysis will give you the clue that what is the location of post translational modifications. Along with the sequence level information, you can also look for the structural information from these fragments. So that fragmentation patterns can also give the insights into a secondary and tertiary structure of the peptides. So you'll also be able to look at the structure, that what could be the structure of a particular peptide. Now we'll look into different fragmentation methods. So till now, is everything clear? Are you able to follow? Okay, thank you. Yeah, so now we'll look into different fragmentation methods. So previously we have not discussed them. So the first method, which is most by default used, is the collision induced dissociation. So what happens, what is the concept behind this collision induced dissociation? The precursor ions, they are accelerated and they are collided with an inert gas. So in that case, inert gas could be either helium or nitrogen, and there is a collision cell. So you can also look it in the diagram which is present below the table. So the first thing is the collision induced dissociation, in which it is shown that how the precursor ions, they are accelerated, they are passed with the speed, and then they are collided with the inert gas. So this collision leads to fragmentation. So the collision with the inert gas is leading to the collision. This process is known as collision induced dissociation. And the characteristic that what kind of ions are produced, so it produces predictable and often sequence specific fragments. So it will lead to the generation of B and Y type ions. And normally, if you have seen the week 4 lectures, then you have seen that we consider the B type ions and Y type ions because they are cutting the peptide at a proper bond which is present between two amino acids. So that peptide bond, when it is broken, then it gives us a complete information of a single amino acid. So in that way, B and Y type ions are very important while looking at the spectra, because they are helping us to analyze the protein sequence. That's why this collision induced dissociation, it is breaking the bonds at that peptide bond site. Hence it is quite useful to use it and to compare the peptides and identify the peptide sequence. Then comes higher energy collisional dissociation. So this is also similar to CID, but as the name suggests, that higher energy. So in this, higher collision energies are used. So what happens, that in collision induced dissociation, the collision is not that strong, but in higher energy it will be very very strong. That means it will be breaking the fragment into more smaller ions. So in that way, and in higher energy collision also, if you see in the figure below, you will see that it is performed in a different or separate collision cell. So collision induced dissociation is happening in a collision cell, but in the higher energy collision dissociation, it is happening in a separate cell, because energy is quite high, and it generates more extensive fragmentation with a broader range of fragment ions. So why this range is broad? Because higher energy is used, so multiple fragments will be formed, multiple smaller fragments. And if fragments will be smaller, then ultimately the resolution will also be high. We will be able to distinguish between multiple peptides. So in that way, this fragmentation method is used. Then comes electron capture dissociation. So as the name suggests, electron capture. So multiple charged cations are used, which captures the low energy electrons, and then after capturing those electrons, because that peptide contains of multiple bonds, so if electrons will be captured, it will lead to the cleavage, because we know that the bonds are formed by the association or the sharing of electrons or those covalent bonds. So after removing the electrons, the bond will break. So in that way, in this case, fragmentation is happening because multiply charged cations, they are capturing the low energy electrons, the bonds which are not that strong. So it will lead to the cleavage of peptide backbone, and it will result in extensive fragmentation primarily of the peptide bonds without breaking the side chains. So in electron capture dissociation also, we will be breaking primarily at the peptide bonds. It's not saying that only peptide bonds, but most of the cases it will break at the peptide bonds, and the side chains will be intact. So this method will also be helpful while analyzing the peptide sequence. Then comes electron transfer dissociation. So it is also similar to electron capture. So in electron capture, you are capturing the electron. In electron transfer, you transfer the electron. So in this case, ion captures the electrons from the reagent anion. So there is a reagent which is anionic in nature. Previously you are using cations. So now in this case, the ion which is already present, it will be capturing the electrons from that reagent. So in that way, electron transfer is happening, and it will generate the complementary fragmentation patterns to the CID and ECD. So these are the short forms of the collision induced dissociation. So now if you know that there are complementary peaks which you see, so when you plus the B and Y ions which are present at a symmetrical distance from one another, so their sum will give you the mass to charge ratio of the entire sequence which is present. So in this way, electron transfer dissociation, it will generate the complementary fragmentation patterns. And then comes photo dissociation. So as the name suggests, photo, that means it is using some kind of light. So whenever UV light or infrared light is used for fragmenting the ions, Okay, voice is again breaking. Um, I think there is some network issue today, maybe that is the reason. So is it fine now? Okay. Okay, then so till now you heard everything, right? You were able to follow till electron transfer dissociation?
Yes, ma'am.
Okay, thank you. Yeah, so now comes photodissociation. So in photodissociation, in the presence of light, whenever the dissociation is happening or fragmentation is happening, so in case of light, we talk about the UV light or the infrared light, it causes the fragmentation of the precursor ions. So this is known as photodissociation fragmentation method, and it generates the fragments through absorption of photons, because light is in the form of packets, energy packets, which are known as photons, and it is typically used in the MALDI mass spectrometry. So these are the methods, and it depends that which type of mass analyzer will be using these things. So that collision induced dissociation, it is very commonly used and majorly used by the ion trap and quadrupole instruments. So the instruments which have these type of ionization methods, so they will use the collision induced dissociation technique to fragment the ions, whereas in case of MALDI it will use the photo dissociation, because in that you see that they use the laser light to dissociate into different fragments, so dissociate the fragments into ions. So in that way these are the fragmentation methods, and this figure explains it all. Then comes if you look at the right side, so this tells about the bonds, so where the particular fragmentation method, how it breaks the bonds between the two residues. So in this case, if you can see, the CID is breaking the bond at the peptide bond site, so C double bond O and NH. So this C double bond is the C terminal carboxyl group of the first amino acid, and this NH corresponds to the first amino terminal, so first NH2 group, which is present towards the N terminal of the amino acid. So in that way, this, if we break at this point, then it will separate the peptide into two different amino acid sequences, or whenever the bond will be broken at this site, so it will cleave, it will not disturb the amino acid which is present, it will be at the terminal position, it will be cleaving. So these types of fragmentations are quite helpful when we have to infer the sequence of the amino acids which are present in the peptide. So that is the reason that why this is famous. Similarly, in this also I have shown that what are the different ions which are generated. So whenever you say A, B, C, so these ions are towards the N terminal, whereas if we call X, Y, Z, so these are read from the C terminal. So the number which is written next to this, for example Z N, Y N, X N, it tells you that how many number of amino acids will it have. So this is the number of the residues which are present. And similarly in the case of ABC also, and most important for our concern, the ions are B and Y ions, and we have discussed why it is important. And in the figure below I have shown that these I have simplified it into the form of amino acids. So whenever there is a long peptide containing n number of amino acids, and this is the first and second amino acid, as I have told you that there is a bond between the carboxyl group of the second amino acid and the amino terminal or NH2 group of the nth amino acid. So this fragmentation will lead to the generation of B and Y type ions. So whenever there will be the transfer of electron, so in this case you can see there is a neutral lost, in which what is happening, the NH2 group, it is transferring its electron to the CC bond, and in that way CC bond is converting to C double bond C with double bond O, which was previously present. Now this NH3, so the one electron has gone to the CC bond, and another electron which was present, it came to the nitrogen, so it has increased its valency to NH3 positive. So in that way there is a neutral lost and Y ion is generated. So in this way Y type ions and B type ions are generated with the neutral loss. So we don't have to go into the detail, this is just an overview that how different fragments are generated and what is the transfer which happens. So in this I have given the information about the mass to charge ratios, that how you can compare. So different amino acids have different mass to charge ratios. So by looking at these mass to charge ratios, you can infer by comparing different peptides. So the peptides, when they are converted into fragments, so they will be of so they will be consecutive fragments. So when I say consecutive, it means that one fragment is having, for example, 5 residues, another is having 6 residues. So by distinguishing or by differentiating those fragments, by subtracting that fragment of 6 residues with the 5 residue, we will be able to get the some mass to charge ratio which will correspond to a single amino acid, and we can look at the table and we can say that this particular amino acid is present at this particular position. So we'll also study one example with which it will be more clear. Okay, so Bn refers to the ions which are coming from N terminus, and what we do is we generally add 1 to the residue mass. So that will be the residue mass, and for example in Yn that ion is formed, so it contains H2O along with plus 1. So the residue mass means the mass of the amino acid. So in addition to mass of amino acid, it will also contain H2O and 1. So H2O, for example, you have seen that whenever Y type ion comes, then you add 19 or you subtract 19 from it to get the molecular mass or the residue mass weight of the amino acid, because in N type ions, that ions also contain the water and one charge. So in that way we have to add this to identify the residue which is present. So why that minus 19 you are doing to Y ions to identify the amino acid? Because that 19 is coming from this H2 and plus 1, because H2 oxygen will have the mass of 16 and H2 will have of 2 daltons. So 16 plus 2 will be 18 and plus 1, so 19. So that's why whenever you obtain Y type ions, you subtract 19 from it to get the actual mass of the residue. So this is the reason, and in B case also you have to subtract 1 because it contains the 1 extra. Yeah, so in this similarly it has been shown that Y3 and B3. So when we say B3, it is coming, we will read it from the N terminal. So 3 means it will contain 3 residues. So if you see here ADG, it will be having 3 residues from the N terminal. So B3 means ADG. And when we say Y3, it means it is containing 3 residues from the C terminal. So it will have a peptide sequence of EVR. And similarly their masses have been added 71.04. So this is the theoretical mass to charge ratio which we could obtain. So with this table they have summed it up and they have looked that this should be the spectra which should be generated, and now when they will look at the original spectra which is generated by the mass spectrometer, so they will be able to identify. So this is just an example to show you that how the spectra will, so to what the spectra will correspond. Okay, so why we are adding this? Because whenever we are doing ionization, so there are some bonds that are being broken and there are certain ions which are present, because that peptide is not, there is some means media, or you can say there is something else in the environment also which is present during ionization. For example, in the previous also we have seen that there are different fragmentation methods. So in some fragmentation method you are passing them through inert gas, or either you are throwing some light, or in the, I'll show you. So for the case of this collision induced, you are passing, you are colliding it with the inert gas, and then in the case of electron capture and electron transfer, you are using some cationic cations or some anionic reagent. So in that case the reagents which are present, they will also be helping to break the bonds. So whenever they are going to break the bonds, they will be transferring something. So in that case, whenever the ions will be formed, they will be containing some extra molecules which are attached to them during the fragmentation. So that's why along with the residues there will be some other ions which are also attached to them. So due to which we need to subtract this 19 from the Yn type ions, because along with the residues it is also having some water molecules, because electron is also transferred. So we so in that way we also have to subtract it to get the exact weight of, or the exact mass of the residue. So that is the reason. Yeah, I'll share the video if it is available. Okay, don't worry. In the slides only I attach the video link so that you can understand. Okay, now this is also another table in which I have shown that, so that because this table is just for your reference, so whenever you will be observing the spectra, so that you can easily compare, you can identify that what would be the mass. And I have also given the composition so that you can understand that how this variation in the combination, or the how the means how this composition is affecting the mass. Because as I have talked earlier also, that if there will be more number of atoms, for example more number of carbon, nitrogen, hydrogen are present, then it will subsequently increase the molecular mass. So in this case, if you look at the composition, so whenever the composition will increase, the smaller atoms which are present in this particular residue or the molecule, so it will change the molecular mass, it will increase the mass if more residues will be more atoms will be present in the particular residue. So when we say residue, we mention that residue as a amino acid, and when we say atoms, so in residue there will be multiple atoms, because amino acid is made up of carbon, nitrogen, hydrogen, oxygen, so all these things are present, and in some cases we also have sulfur as well. So these all are the atoms which combine to form this residue. So in that way, different amino acids have different composition of these atoms, due to which they have different atomic masses or molecular masses. So in that way, this composition will also help you to differentiate that how they have certain masses. And in this I have given the ion types and their neutral molecular weight of the fragment. So one is the molecular weight, N is the N terminal, the nitrogen which is present towards the N terminal, and CO refers to the C terminal carbon and oxygen bond along with the hydrogen which is attached. So in that way these ions are generated. So this is just to show you that whenever you generate ions, those ions contain some extra charges and some extra atoms attached to them, not just the residues only. So due to which these ions will have some extra molecular mass attached to them. So to identify the residue mass, we have to subtract the mass of the attachments. For example, in the A type ions, the H2O is present, so we have to subtract 18.0106 to get the exact molecular mass of the A type ions. So similarly, I think you have also seen that the difference between the A and B is that you have to subtract minus, B is equals to A minus 28. I think that you have seen. So all those things you can look at this table, that means how those ions are different from one another. So I won't go into the detail of them, but this is the reason that if these are residues are present, that's why we are separating them. That's why we are separating that mass, so that we can get the exact mass of the residues and not the other atoms which are attached to that particular fragment. Okay, so then comes the digestion, different methods of peptide digestions. So firstly, when we obtain the proteins, we are digesting it into peptides, then we are performing the ionization or fragmentation. So for digestion we have looked at trypsin. There are different enzymes which are also used for the digestion of protein into peptides. So this trypsin, this I am giving you this information because when we will be analyzing the spectra, then through these methods we get some kind of hints. Because if trypsin is used in the cleavage, you will be very sure, because trypsin cleaves at the C terminal basic residues, which will be mostly arginine and lysine. So whenever digestion is done with the trypsin, so you will be sure that its C terminal will always be either arginine or lysine, because it cleaves at that position. So after the arginine or lysine, it will cleave. So in that way this is helpful. Similarly, if the digestion happens with chymotrypsin, so it cuts after the aromatic residues. So aromatic residues are present in the aromatic residues are the phenylalanine, tyrosine and tryptophan. So whenever chymotrypsin is used, so you will be pretty sure that towards its C terminal we will see either phenylalanine, tyrosine or tryptophan. So this is just like a hint which you can get if you have the idea that what is used in the digestion. Similarly, other enzymes are also present, such as pepsin, elastase, V8 protease. So they have some specific cleavage sites which will help us to identify the terminal residues or terminal positions what could be present. So this is in the bottom figure I have given the example that whenever trypsin digestion is happening, so if this is a long stretch of a protein, so the peptides will contain either lysine or arginine towards their terminal. So this is all about this trypsin. And now in this example, this is also to show you that how the different tandem mass spectra is working. So firstly, there is a protein. After protein is obtained, we will reduce it, we will perform alkylation, and we will also perform the trypsin digestion to obtain the peptides, because we cannot analyze entire protein through mass spectrometry. We have to break it into peptides. So for that we will be using the trypsin digestion, and there could be some other digestion methods as well. If we don't use trypsin, then we can use some proteases which breaks the bond between the proteins. So in that way the digestion will happen. Now we will perform either liquid chromatography separation methods 1D or 2D. Now we will pass it through the mass spectrometry. So in MS1, if you see, there will be multiple fragments which are present. So in this, if you can see this figure where MS1 is written, so in that there are 3 different types of peptides which are present. So why we use multiple MS? Because now what we are doing, we are selecting a particular peptide to pass through the collision cell and ultimately reach the MS2. So in this way, one level of selection is done. We have selected some mass from the peptide of interest. Now this peptide of interest will pass through the collision cell. In collision cell it will be further fragmented, and now MS2, the second mass analyzer is present, it will sequence this peptide of interest. So now this peptide is further fragmented and we will be able to identify the sequence of this particular peptide. So due to this reason we use tandem mass spectrometry, because we get the advantage of selectivity. So we can select at the first level that which peptides to be passed. Now we will perform one exercise. So this is a quite simpler exercise, so that you can understand that how we infer the sequence of the peptide by looking at the spectra. So whenever you see spectra, it tells you about the mass to charge ratios as well as their relative abundance. So relative abundance is helpful when you are having multiple samples and you want to compare that what is the difference between them, which peptides are more expressed, which proteins are differentially expressed. So for that reason we look into the relative abundance. So in this case, this is the sequence, and we are very clear because this is a trypsin digestion, so it will be having the lysine at its C terminal, and for your simplicity leucine is given at the N terminal. So I hope you are aware about the one letter code of amino acids. So L stands for leucine and K stands for lysine, and in this table you can take help of this table to identify the residues which will be present. So in this case, as you can see, so simply let's start with the peptide sequence. So there are 4 spaces which are present and 2 are already given. So that means the entire peptide is constituting of 6 residues or 6 amino acids. So Y5 and B5 both are present, so we can use both the informations. We can use both B type ions as well as Y type ions to get the information. So now let's begin. So whenever we'll say Y6, so Y6 means it is containing 6 residues from the C terminal. So Y6 ion, if that will be given, so that is not given here, but if it has to be given, so that will correspond to the molecular weight of this entire peptide sequence. But in this case they are not given that, so Y5. So when we say Y5, that means 5 residues if we start the counting from C terminal. So that means this Y region highlights that Y5 will generate the ion from K till the second position. It will not include L in it. Y6 will include K to L entire peptide sequence. Y5 will correspond to K till second position, not including L. So now as we don't have the Y6 mass, otherwise we'll be subtracting the Y6 and Y5 and we'll get the molecular weight of L if we don't have the L, but we already have leucine at the N terminal, so we don't need that. So now we'll move to the Y4. So now if we subtract Y5 from Y4, so it will give us the sequence which is present at the second position. So am I audible? Okay. So this is means if you won't get anything in this example, then please let me know. I will try to go slowly so that you can understand. So as I mentioned that when we are saying Y6, so it is including all the 6 residues. When we are saying Y5, so please make this thing clear in mind that we are reading one by one from the C terminal position. So in case of Y5, it will contain all the residues except L, so minus L, minus 1 position. Similarly, when we go to Y4, so it will contain all the residues except 2 residues, so L, N are not present in Y4. So now we will be subtracting Y5 from Y4. So Y5 is present till second position, Y4 will present till third last position. So when we subtract them, we will be able to get the molecular weight of the residue which is present at the second position. So now the difference will come out to be 114.08. So in the right side in the table, if you can see the third position, the third residue is asparagine. It corresponds to the mass of the residue to 114.08. So in this way we will fill N in the second position. Now we will look at the Y3 and Y4 because that will give us the residue molecular weight of the third position. Okay, so now if you can see Y4 and Y3, if we subtract them, we will get the difference of 99.08. and if we will refer to the table at the right, so in the last position you can see valine corresponds to the mass of 99.08. so in this way third position will contain valine. Similarly, now we will move from Y3 to Y2 and we will calculate their difference. So their difference is 186.18, and in the table if we see third last residue, it is tryptophan, which corresponds to the mass of 186.18. so we will fill tryptophan in the fourth position. Similarly, now we will subtract Y2 with, so we don't have Y1, so we won't be able to separate that. Now let's see what we can do. Yeah, so now till Y2 we have 185.26. so Y2 is 185.26, and we already know that Y2 will contain K and this particular residue which is present at the fifth position. So now if we subtract the mass of the K from 185, it will give us the mass of the residue which is present at the fifth position. So we will subtract, so K is the lysine, it has the mass of 128.18 if you see the table. Now we will subtract 185.26 from, so we will subtract 128.18 from this 185, we will get 57.08 which correspond to glycine. So in that way we will fill glycine. So in this way we are able to identify the sequence of residues which are present in this peptide sequence. So this is the way how we analyze the spectra. So in this example I have shown you that how we can read the spectra from the Y side, so from the C terminal. So we can do same by, okay, so if K is not given, then you have to look for some other alternative. So in that case maybe Y1 peak will be given, or maybe the in most of the examples they give you the complete mass of the peptide sequence. In this example they haven't provided you, but in most of the cases they will provide you the mass to charge ratio of the entire sequence of the peptide. So in that case it will be helpful, that will tell you the entire mass and you will simply subtract that. So entire mass will correspond to the Y6. So in that way Y6 will be available with you. So yeah, so in that way you will be able to identify both at the L side also and at the N side also and the C terminal also, and you can also use the B type ion information also. So in this case I have only used the Y type ions. So similarly we will look that how we can also use the B type ions to infer the same sequence. So similarly if some peak is not available for the Y type ions, then we can use the B type ions, and if B type ions also not available, then we can also take the help of A type ions or other ions, because we have seen there is a relationship between these ions. So by subtracting some fragments we can be easily getting the mass of the B and Y type ions. So those things can be done. Okay, so now let's begin by taking the information of B type ions. So in this case if you see, B6 will correspond to the entire peptide sequence. So when we will begin from the B1, so B1 means it will only have L, that is leucine, and when we say B2, it means it will have both leucine and the second position residue which is N, we know, but now we don't know. So what we will do is we will subtract B2 from so we will subtract the value of the mass of the leucine from this B2 mass, which will be 114.08, and which corresponds to the N, which is asparagine. So we will keep N here. So in this case I have already written because we have analyzed from Y6. So in this way it will correspond to N. Similarly we will compare between B2 and B3. We'll subtract them. We will get 99.08 if we subtract this mass to charge ratios, and we'll get 9.08, which corresponds to valine. So third position we'll get valine. Similarly we'll subtract B3 and B4, and now after subtracting them we get the mass of 186.18, which corresponds to tryptophan. Similarly now B4 and B5 are present, so we'll subtract the mass among both of them. So we'll get mass of 57.08, which corresponds to glycine. Now similarly in this case also we do not have the information of B6 ion, but we already have K at this position. So in that case we do not require any further information because we already have the terminal positions. So in this way, by using both the B and Y type ions, we can infer the sequence of residues which are present in the peptide sequence. So till now everything is clear. Okay, so this was quite simple example, but in reality the mass spectra is quite complex and quite weird to analyze. So for that you need that exercise and experience to study the spectra. This is just a simple spectra for your for your understanding, so that you can understand it in a simpler way, and we do not have to go into the detail because you won't be given the spectra to analyze the peptide sequence, so don't worry about that. And now how to evaluate the protein mass spectra. So this slides contains the information that what things you should keep in mind, that whenever you are observing some kind of spectra, so whether it is genuine spectra or whether you should consider that peak or not consider. So first criteria is to look at the molecular ion peak, and that peak corresponds to the intact protein or M positive or the charged state. So now there are two things, that whenever you can accept that peak and whenever you can reject that peak. So if the peak is clearly present and matches the expected molecular weight, then only you will accept that peak, otherwise you will not consider that peak, because there are multiple peaks which are present in the spectra. So this is just telling you that which peak you should consider, and if peak is missing or weak or doesn't match the expected molecular weight, you will not consider that peak. So in the lectures also you have seen that if that sir was telling you that there are some peaks which are very closely present to one another, their difference was hardly 20 or 28, means their mass was varying by only 28 units. So that thing, in that case we are not giving any means we cannot subtract them, we cannot give them the Y type ion. So in that scenarios we should ignore that peak, because they are closely means they are present at the closer positions and they cannot correspond to two different residues. So in that case we can ignore those peaks because they are not matching the expected molecular weights. Now comes the charge state distribution. So there are series of peaks with the different mass to charge values for various charge states. So in that case the charge states also, there are some ions which are doubly charged and some are singly charged, so those peaks are also present. So in that case you have to be very careful looking at their masses, because if those peaks are coming at the mass to charge ratios, so if it is doubly charged, so its mass will be half, because mass to charge, so you are dividing it with the charge. So when it is singly charged, its molecular mass will correspond to the exact molecular mass of the residue, but in certain scenarios what you see is that the mass is half of the molecular mass which you are expecting. So in that case the thing what can happen is that your molecule or your ion is doubly charged, so now you are dividing it with 2, so that's why your mass is half. So in that case charge states are well defined and consistent with its protein's size, and if the charge states are irregular and poorly defined, so you have to reject those states, you have to reject those peaks, you don't have to consider them. Then comes isotopic pattern. So cluster of peaks spaced by 1 dalton per charge state. So in certain cases what you see is that two peaks are very close, they are just separated by 1 dalton. So those peaks can also arise due to the isotopic differences. So the cases where these peaks are clear and match the expected pattern, in that case you can accept that peak, otherwise you should reject that peak. Then there are signal to noise ratios. So if signal to noise ratio is higher, which tells you that signal is more and noise is less, so in that case you can accept that spectra. Whereas if there is low signal to noise ratios, then you can reject that spectra. Then comes peak width and resolution. So we have also discussed that if peak width is large, which means the resolution will be poor. So peak should always be sharp, so that we can distinguish between the two peaks which are present next to one another. Then comes background peaks. So background peaks are the peaks which are other than the residue peaks, because there could be some contaminants or known protein sources which are present, because there could be some contamination due to which you will observe certain peaks. So you should ignore those peaks. If there are minimum background noise or there are few irrelevant peaks, then you can consider that spectra. But in certain cases, if there is a lot of peaks, if you are observing it towards the terminal region or in the spectrograph, in the end if you see multiple peaks which are means the spectra should always be present in a Gaussian distribution state. So if it is like starting with the low, it is rising and then it is decreasing, and in the end if you see sudden larger peaks, so that means there is some contamination which is present. In that case you should ignore that spectra, or you should ignore those peaks while taking the measurements. And then reproducibility. So reproducibility means that if you are taking the reading multiple times, so the spectra should be same which you are obtaining. If it is varying, then you also have to take that into consideration, that why it is varying. Is there something with your sample, or is there something with your is it some technical error? So all those things, they should be taken care of while comparing and making your hypothesis and testing it and analyzing it. So in this I am showing that how the contaminations are observed. For example, if this is your spectra and this is your relative abundance among different mass spectra, and suddenly you see that there is a very high intensity peaks which are observed towards the end of the column. So what is happening here? This could be a contamination, because whenever you are performing the chromatography, and whenever in the chromatography also whenever the illusion happens, so if multiple peaks are coming in the end, that means there is something other than that particular peptide, something else is also present which is coming up in the spectra. So which means there is contamination, and we should avoid these peaks, we should not consider these peaks. And when we talk about the peptide mass fingerprinting, so peptide mass fingerprinting is nothing but it is simply a term which is referred to as identifying the residues which are present in the peptide. So how you can, because different peptides are means you can distinguish between peptides by their mass to charge ratios. So due to this variable mass to charge ratio, you are able to distinguish between these peptides, and you are able to infer the residues which are present. So in peptide mass fingerprinting, you are simply able to identify the different masses of different peptides, and you are able to associate those peptides to their peptide sequence. So in this example, it is shown that there is a 2D gel from where you have performed in-gel digestion, you have performed mass spectrometry, and these all the peptides. So in the left side, if you see, there are multiple peptides which are shown, and these are their masses. So mass to charge ratios, these are their mass to charge ratios. Now what you do, you already have a database in which already human proteome sequences are present. So I am taking example of human proteome because we are analyzing the human protein sample. So in the database there could be multiple proteins which are present. Now in silico digestion means the digestion which is theoretically performed. So for example, I know I have performed tryptic digestion. So what I will do is that in the experiment I have performed tryptic digestion, I have obtained certain peptides. So now what I will do, I already have the database of human proteome. I will simply look for the protein sequence. I will cleave it by looking at the positions where the K or R is coming. So wherever lysine and arginine are coming, I will chop off. I will obtain that sequence theoretically. That is not broken by the trypsin enzyme. I am doing it theoretically. I am breaking my protein sequence into multiple peptides because I know that it will cleave at the C terminal end of the um, lysine and arginine. So in that way I will generate multiple peptide sequences. Now I already have a table which tells me what could be the expected mass of that particular residue. So in that way I will sum up all the residues which are present in the peptide sequence. So I will get the list of the theoretical masses of each and every peptide coming from multiple proteins. Now what I will do, from the experimental peptides I'll take their experimental masses and I'll compare it with the theoretical masses which I have generated. So in this example, if I am comparing, so these peptides are more comparable to my second protein, so for which I have generated these theoretical peptides. So in that way we are able to identify the protein sequence as well as the peptides from where this means. We can also relate the peptides, that what could be their sequences, because we already have the database. So in this way we are able to identify the protein which is present in the sample, because in the sample we don't know which protein is present and what is the abundance of that protein. So in this way comparison can be performed by comparing both the experimental as well as theoretical peptides. So I hope this is clear. Okay, thank you. Now we'll look into different issues in mass spectrometry. So these issues are not with the mass spectrometer, but these are the issues depending on the instruments which are used, or depending on the conditions in which you are performing experiments, time periods, or the gel which you have prepared. For example, there are different things which could bring about the disturbance in your experiments. So first thing is the ionization variability and instrumental noise. So for example, there is variation in their ionization efficiency. Or what if I am using different mass spectrometers for my analysis? For example, I have taken normal person sample and a cancer patient, and I am using different types of ionizers to perform the ionization and different detectors. So in that way I have used different methods. So in that way from their results I cannot exactly infer, because there is a difference in the techniques which I used, different in the instruments which I used. So in that way the noise which can come through instruments or the ionization variability, so in that way there will be difference in the samples or the identifications which are done. So in that case we are not that much sure that whether the variations which are coming in their peaks or the proteins which are for which the abundance has been observed, whether it is due to the error of my machine or whether it is exactly true, that it will be observed truly. So that is one of the issues. Second is the sample preparation errors and degradation. So sometimes what happens is you are preparing your sample, but some contaminants come up, or there is degradation of the analyte in which you are performing your experiment. So during the liquid chromatography, if there is some issue in the movement or in the analyte concentration, so that errors can also come up, which could also lead to the inconsistency in the spectra. For example, we have seen that there is some contaminants. If the contaminants are present, so huge number of peaks are observed towards the C terminal, so they are just the contaminants, so we cannot observe them. Then comes resolution and calibration drift. So if there is insufficient resolution, that means if the width of the peak is large, or we are not able to distinguish if there are multiple peaks, complex peaks which are present in the spectra, so that is also one of the reason that we should avoid it. So in that way, whenever we are performing liquid chromatography or we are using multiple mass spectra, so we should be careful that we should try to improve the resolution. So we should not run behind that we have to quickly perform the experiment. We have to think first of all we have to think that proper separation need to be done, proper gradient has to be used, which could distinguish clearly between the different types of fragments, so that in the end we will be able to identify the spectras and we will be able to distinguish among spectras. If we will not perform steps properly, then what we will see, that we'll see lot of spectra lying on one another. There will be overlapping spectras which we will not be able to distinguish between them. So that thing can come due to resolution and calibration drift, because in calibration you are not properly calibrating over time, so that can lead to overlapping peaks. And then data interpretation challenges. So if the spectra is complex, then ultimately it will also complicate your analysis. Your interpretation will not be good. You will be confused that what ionic peak you are observing. So because you also have to label that this should be firstly you have to see that what is the entire mass of the fragment, then you have to label that what would be your Y6, sorry, it depends that how many residue long peptide is used. So you have to label them that this would be my Y6 ion, this would be my B1 ion. So in that way, that if the spectra is complicated and your interpretation is wrong, then in that way your analysis will also go wrong. So these all things are the issues which you will observe in the mass spectrometric experiments, and you have to take some measures so that you can address these issues. So first thing is the initial assessment. Whenever the data is acquired, data is obtained, you have the raw data matrix, you have to normalize it, because if multiple experiments are performed, there could be different units in which that experiments are performed, so you have to bring them to a common ground, you have to normalize them. So data matrix will be normalized. Now there could be some batch effects, so you have to correct those batch effects. And now after correcting the batch effects, batch corrected data matrix will be obtained. Now you have to perform the quality control, and in the quality control you look at those interpretations or those means the data which you are having now, so you have to check that data, that whether it is accurate or not, whether there are missing values, so you have to impute them. You have to see that whether the data which you are obtaining, whether it is relevant or not, so that comes under quality control. Then protein quantity inference, so because abundance is given by the mass spectra, so you will be able to distinguish among different peptides, and if number of peptides are more and they are corresponding to a single protein, which means that protein is overexpressed in that sample. So in that way protein quantity inference can be made after batch corrections, and then statistical analysis can be performed to make some other hypothesis and to test that hypothesis and accept it. Now let's talk about the batch correction. So batch correction is very important step which can, so batch means this particular error can arise due to some technical variability which can be introduced by difference between the sample batches and experimental runs. For example, it is advised that whenever you are performing experiments between the normal and disease samples, so in the one day you have to randomly perform those experiments. You should not do that ki for few days you are only performing experiments of the normal samples, and then after 3-4 days you are performing experiments for the cancer samples. So this thing you have to be make sure that you are performing them randomly. You are doing random sampling from both of those samples. You are taking the samples and you are performing the experiments. So if you are not doing in that way, so there could be some batch error, because it depends that in which conditions you have performed your experiments, so that could introduce the technical errors. So to remove them we have to perform batch correction. Now there are some key points which you must know. So what is the purpose of this batch correction? So batch correction is important to adjust the systematic biases and variations which arises from the different batches or runs, ensuring that data across all the samples are comparable. And I think in the lecture only they have mentioned that, for example, in the first month you have performed some experiment, then you go for a leave, and after 3 months you came up and you performed another set of experiments, and now you are going to compare the experiments performed in the first month with the experiments performed in the 5th month or the 4th month. So in that way they will show some kind of variations. So you have to make sure that that variation is not because of your time of experiment in which you have performed the experiment. So that could be a technical error, so you have to check for that. So you have to adjust for that, you have to perform batch correction. So that is the purpose of this batch correction. And then if we talk about the sources of variations, that what could be the variation sources which are leading to this batch errors. So it could be differences in the sample preparation, instrument performance. It could also be possible that your instrument is not working properly now, so that could also add up to the error. Or maybe the samples which you are preparing, they could be different, and analysis conditions in which you are performing the experiments. So there are different conditions which could influence your batch, so which could bring about the batch errors, and we have to correct them. Now how to deal with these batch errors, what can we do? So first thing is normalization, which means you can adjust your data to a common scale using the techniques like quantile normalization, internal standards, or reference points. So these all things we should do. Normalization we should do scaling. Scaling is like you apply a scaling factor to align the data across batches, often based on the subset of features or common internal standards. And then third method is the statistical correction, in which you use statistical models to adjust the batch effects, such as linear models, combat, and other empirical based methods. So these all are the methods which are used to deal with the batch correction. And in this table I have shown different methods, what are their purpose and what they does, and what are their strengths and what are different limitations attached to them. So first of all, combat, which is majorly used, and this is the most robust method which is used for the batch correction. So when we say combat actually stands for the batch correction method, and it corrects the batch effects using the empirical base approach. So there are some statistical approaches which these methods use to normalize your data and then to remove the batch effects. So it uses the empirical base approach. It adjusts for the batch effects by modeling the batch specific parameters and leveraging empirical base. So in that way it removes the batch effect. It is highly robust and it is widely used in proteomics. It handles the high dimensional data as well. So if the data is high dimensional means if there are multiple features which are associated with your data is huge. So in that case also you can use the combat. So the limitation with the combat is that it assumes that batch effects are additive and can be modeled linearly. So if batch effects are not additive, if batch effects are random or they are not adding up, so in that case the combat will not be able to correct your batch effect. Second method is LEMA, which stands for linear models for microarray data. So it is also used to analyze and correct the batch effects with linear models. And what it does is it applies the linear model to adjust for batch effects in proteomics data, and it is well established. It offers flexibility in handling different types of data. And if we talk about the limitations, so it is primarily tailored for gene expression, and it was not made for the proteomic data set. So we have to perform some kind of modifications and we have to adapt, we have to select certain options when we are looking for the proteomic data normalization, because it was not made for the proteomic data analyzation. Then comes k-bit method, which stands for k nearest neighbor batch effect test. It also assess and correct the batch effects by maintaining the data distribution, and it checks for the batch effect by ensuring that the batch distributions are similar. And if we talk about the strengths of this, so it preserves the integrity of data distribution and it is effective for the larger data sets. But the limitation is that it is relatively new and less established, so it may require fine tuning because there are multiple parameters which we need to specify when we are removing the batch effects. So in that case this is not well defined because k-bit is quite new, so we are not used to it. We do not know what should be the ideal parameter which we should use. So in that way k-bit is not that much used as compared to combat. Then comes SVA, which is surrogate variable analysis. It also adjusts for the batch effects and other unwanted variations. It identifies the surrogate variable to model and adjust for the unwanted variations. So these all methods, they are kind of based on some statistical techniques to identify that what is that particular constant value which is introduced by the batch effect. So they simply adjust that value, they simply either subtract that value and try to normalize your data so that it does not have the batch effect. So similarly SVA is also the same. And one thing which you should take away from this slide is that combat is most robust method and it is majorly used, and it uses the empirical base approach to correct the batch effects. Now what are the steps in the correction, because as we are talking about the batch correction, so there are multiple steps which are used. So first thing is to identify the batch effects. So first step in which we detect and quantify the variations which are attributable to the batch differences. So if there is no batch effect or there is no difference between the two samples, we cannot figure out, so in that case we cannot go for the batch correction. So firstly we need to identify that what is the batch effect. Then we have to choose the correction method, that which method we want to use. So depending on the nature and extent of the batch effects, so if the data is high dimensional or if batch effect is small or if batch effect is huge, so in that way we could select any correction method depending on our requirement. We should apply the correction, we should implement the chosen method to adjust the data so that the batch effect is minimized or either removed. And ultimately we validate our results. We will check for the effectiveness of correction by comparing the distributions before and after applying this correction. So by that you will be able to know that whether your method is performing better or not. So in the later slides we will see that how two dataset varies before normalization and after normalization, and what normalization is actually doing, and how you can analyze that whether your normalization is doing a good job or not. So that we will see. And then if we talk about the benefits of batch correction, so it improves the data quality because it is removing the batch effect. And then it is also enhancing your comparability, because previously you were not sure that whether that difference was due to the batch effect or that was the real difference between two samples. So now you are enhancing your capability because you are removing the batch effect, so you will be able to compare two samples in a better way. And considerations, so there are some things which you need to be careful. First is overcorrection risk. So you should always avoid the overcorrection, which can potentially means disturb the true biological differences. And then method selection, so you should be careful while selecting the method, that which batch correction method you want to use. So if we talk about overcorrection, so this was also mentioned in your week 4 only, that in certain cases whenever you are observing some samples in which the some proteins are overexpressed and some are not that much expressed, so in that case there is some kind of selective expression of certain proteins. It is not like uniform expression. So in that case, whenever we are performing the batch correction, so it might lose the data of the, you can say it as outliers. It will refer that particular overexpression as the outliers and it will ignore that data during correction. So in that way, the potential biomarkers which could have been identified otherwise, they were ignored due to this batch effect correction. So we have to be careful while using this batch correction so that it does not perform the over corrections which is not needed. So all those things have to be kept in mind. Now there is one literature on this. So it was about the batch effect removal, the using the deep learning method, and it was used for the MALDI MS based metabolomics. So in this plot they have shown that how before calibration the peak was obtained and after calibration how the differences you can observe. So calibration is kind of batch means batch correction after removing the batch effect. So in the left side I have explained this figure. So if you see figure A and B, if you compare among both, so before calibration the samples were obtained from different batches. So these different batches were performed, so these experiments were performed on 2 different days. So samples were collected from 2 different batches and they are clearly separated by the batch effects which can hinder the classifications. So now they are separated because of the batch effect. So we need to perform the batch correction, we need to bring them to the same comparable positions. So after calibration the batches distributions become intertwined, indicating the successful batch effect removal. So previously they were spread, but now after performing the calibration or after performing the batch correction, now they got condensed. So we are kind of condensing that information, we are trying to normalize the spread. So now the spread is normalized, so we will be able to capture them in better way. We have removed the batch effects which could arise due to the days in which experiments have been performed. So this shows that our batch correction method is accurate, it's correct, because it is trying to bring both of them to the single position, it's trying to merging them to one another. Now in the figure C and D, if you compare, so first thing is before calibration class 0 and class 1, and in this after calibration class 0 and class 1. So first thing was at the level of samples. Now second thing is that you are also able to categorize these samples, because now you have calibrated them. Now the samples are grouped according to their true labels rather than batches, so demonstrating improved classification and feature space alignment due to batch effect correction and network discrimination. So when 2 different batches they have used, means 2 different days 2 experiments were performed, so when it was represented in that way, so there were some batch effects. After removing those batch effects on the basis of day, now the data has come into the common ground, there is no batch effect, so those days are not affecting the output or the outcome. Now they are able to calibrate them and distinguish between the labels. So now in this labeling, in the C and D figure, they are not labeling them on the basis of the day in which experiment was performed, but they are labeling it and they are comparing among both the categories and they are able to distinguish them clearly. So in this paper they have talked that how the batch correction can help you to firstly to remove the batch effect and then clearly distinguish between 2 different samples and be sure that that difference is not due to the batch effect, but that difference is the real difference between the 2 samples or the proteins which are expressed in the 2 samples. So this is all about this method. Now let's talk about data normalization, because it comes under batch correction. So in batch correction you go for the data normalization. So data normalization is a critical step which is used in the pre-processing of the raw data, because raw data is raw, it is not having any proper information attached to it, you are not able to analyze it, so you have to normalize it, and you have to ensure that the results are accurate, reliable and comparable across different samples and experiments. So a little example I can give you, like for example in class you have 10 different subjects, and in 10 different subjects each and every teacher decides that I'll take test from 10 marks, some decides I'll take test from 20 marks, some decides I'll take from 30 marks. So now they have different scoring patterns. Now how will you judge the performance of each and every student? So in that way we have to normalize that. So for each and every subject we normalize it, we take a single percentage of a single person, and then we can compare the students among the class, that what is the performance of each and every student. So that is what done in data normalization, because different samples can have different, means different measures which are taken, different data points which are present in each and every sample. So we have to normalize it, bring it to a common scale, so that we can distinguish, that we can compare those two data sets. So that is the purpose of data normalization. It kind of process the raw data to a useful information. So it adjusts the values which are measured on different scales. For example, in this case the original data, if you see the peaks, it was from 0 to 4, and then ultimately it diminishes. But when we will normalize it, what we will do is we will convert it into the proper spacing, in which we have a 0, +3 and -3 values, and we have the peaks. So we have adjusted it, we have converted it, so that from other examples, other data sets also, we can adjust that it into the normalized form, so that they all data sets can be compared. And it also compensates for the technical variability which might have arisen due to differences in the sample preparation and instruments. So that's why this data normalization is used in the batch correction, because it is removing the technical variability. Yeah, so now the thing is that why we should use data normalization, what it is doing. So firstly it is compensating for the technical variability, because it is reducing the impact which was incorporated due to the sample preparation differences and instrument performances. Then it ensures the data comparability, because it is bringing it to a common scale for all the data sets. So it is helping us to compare the data. It improves the data quality, because previously the data was like raw reads, but now the quality has been enhanced by performing the normalization. And in normalization what else you do is that there are certain values which are lying far away from your data, major values of the data. So in that case you do not consider those values, you remove those values, so that you can get the better analysis, or you restrict yourself to the useful values, you don't consider junk in your analysis. So in that way it improves the data quality. It corrects for the systematic biases which might be generated during the signal detection. Then it facilitates the downstream analysis. Obviously if you are removing all those errors, then downstream analysis will be smoother, because you are removing all the variances which are not of your need, and you are aligning the distributions. Then reduces the impact of outliers. So yeah, outliers are the values which are lying far away from your whole data. So in that case you are reducing, or you are removing that outlier, so that the other data which you have, you could analyze it in a better way. And that outlier could also be due to the contamination, or it could skew your distribution. So normalization helps you to reduce that impact as well. And now I have given certain techniques, and if you want to go through them you can go through, and there are certain formulas. So first is linear scaling. So linear scaling is used when you have the features uniformly distributed across the fixed range. Then z-score scaling, when the feature distribution does not contain the extreme outliers. So in that case you can go for the z-score scaling, and this is the formula. Then log scaling, so when the features conforms to a power law, so if you see power law distribution you can go for the log scaling. Then clipping is like you are fixing some maximum or minimum value, and you are only taking features which are present in that range, so that is known as clipping. So when feature contains extreme outliers, yeah, there could be some outliers which are lying far away from your data, so in that case you can specify that what should be the maximum value. After that you will not be considering the values which are lying far away from that particular max value. So that is known as clipping, and it depends what data you have. how it is distributed. So in that way you can select any of these normalization techniques. And in this table I have also given the other normalization methods which have been discussed in your week 4. So firstly it was centering, that you center your data on the basis of the mean and median. Then you can also go for the scaling, in which you use the standard deviation scaling or mean absolute deviation scaling. In standardization you can go for the z-score standardization, where you try to center your data, and it is standardized in such a way that it has a mean of 0 and values are distributed across 0. So what it is doing across various samples, it is trying to bring it to a common scale. That is the whole purpose of normalization, so that you can compare among different samples which were varying in their scales. And then median centering with MAD scaling, so it also centers by median and then performs the median absolute deviation. Then comes quantile normalization and two component normalization. So in quantile normalization also it adjusts the data so that distribution of protein intensities is the same across all the samples. And in two component normalization also it normalized based on both the internal standards and reference proteins to account for the multiple sources of variability. So we'll look into the detail of quantile and two component normalization in the later slides. But till now I hope it is clear that in data normalization we are trying to bring it to a common scale. That's why we could either look at their mean values, and then on the basis of mean values we'll distribute the other values, or we can perform their median centering. And these are different methods which we generally use so that different samples can come to a common scale for their comparisons. And at the same time we are also removing the values which are of not any importance, which are just the contaminant values, which are the far outliers. So we can ignore those values which are just biasing our distribution. Because when you're if there should be a Gaussian plot or there should be a normal distribution, so due to the outliers that distribution appears to be skewed, because your outlier is lying far away from your values. So in that case it shows that there is a skewed distribution. So to avoid those things we perform normalization so that we can get rid of the noise, or you can say the unwanted values or unwanted data which is irrelevant. So we can get rid of that by using this normalization methods. So you can go, if you want to go into the detail, you can go through this table. And then there was another paper in which also they were discussing about different normalization methods which are used in the mass spectrometry for different clinical samples. And in this I have given the highlights of the paper, that in this firstly they are mentioning that if there is lack of normalization method, comparisons in large scale mass spectrometry based proteomic data sets, so where normalization is necessary in order to remove the unwanted variations. So they are saying that there are not many normalization methods which are present, and these are very important to remove the unwanted variations. So in this case they have taken 8 different normalization methods, which are standardization, quantile sample, single standard, and RUV. So these are multiple methods, you don't have to go into the details, but you are aware of the standardization and quantile normalization and combat. So they are also comparing that combat using the plates as batches, combat using sites as batches, then mean centering, median centering. So these are different kinds of normalization methods which they have compared, and they have identified that what should be the normalization method used and for which conditions which method is better to use. So you can go through this paper in which they have taken the obese human plasma collection from 8 different countries. They have taken their data, and ultimately during analysis they have used these data normalization techniques. So in this okay, so I'll give you example that how the quantile normalization is done. So quantile normalization is kind of correcting the technical biases by equalizing the distribution of the sample values for each sample in the data set. So now I will tell you what are the steps which are performed in quantile normalization. So let's look at the figure, which is these tables which are shown in the left side. So in the A it is showing the samples and the proteins, and there are different batches in which these experiments are performed. So in the B figure, if you see, firstly the table represents the raw data. So in each raw data there are some values which are written. Now in the quantile normalization we have to normalize it because different batches will have different values. So firstly, to normalize it, firstly we have to rank the values of each protein in each sample from the lowest to highest. So sorry, lowest to largest. So let's look at the first column. So in the first column, if you see it's written as 25413. So are you following? Okay, thank you. Yeah, so if there is any confusion during this, so please let me know, because it's quite complicated. If you're not able to follow, if you miss even a single thing, then it will be difficult for you to understand. So now let's carefully look at the row data. So if you look at the first column from 2 towards 3, so 25413, in the first column these values are written. So first step is to rank each value in each sample. So now after performing the ranking, the next table, if you see, they are arranging it, they are sorting it on the basis of, means it is written in the ascending order. Now as I was talking, that in the case of quantile normalization, what we are doing is we are looking at the row data. So now this entire column of this row data, we will firstly look at this, we will try to sort it into a ascending order. So as you can see in rank based data, what we have done for this first column, we have sorted the values in the ascending order. So in this case, if we sort these values, it will be turn out to be 1 2 3 4 and 5. Similarly, for the second column of the row data, we will sort these values. In rank based data, we will be sorted them as 2 3 8 8 10. So these were randomly arranged like this. Similarly, for our third column also, we will sort these values, and similarly for our fourth column also, we will sort these values accordingly. So now after sorting, now we will take the mean of their rows. So in this case, these are different rows which have the rank based data. Now for each row we will take the mean. So in this case, this row's mean will come out to be 3. Similarly, this row will have the mean value of 3.75, and this row will have mean value of 6, and this row will have mean value of 6.5, and similarly this row will have mean value of 7.75. Now what we will do in quantile normalized form, as we have seen, there are 3 steps. Ranking is done, average is calculated. Now the third step is to substitute. So now in this case, these values will be substituted with the mean rank values. So now what do you mean by mean rank? That we will look at the row data. Now we will look at the first column. Now first column's first value is 2. What we will do is we will check this value in the rank based data. So where this 2 lies in the first column, so in the first column it lies in the rank number 2. Now we will see what is the row mean of this column. So this particular value, so its row mean is 3.75. So now we will replace the value 2 with 3.75, which is the row mean, which is the rank row mean. So which means each value has some rank in the column. We will check that rank and we will replace the value with the mean of that rank. So this second rank has mean of 3.75, so it is replaced with 3.75. Now similarly, if you look at the value of 5, it is in column 1, and in case of rank based data, this 5 lies in the 5th position. So now we have to replace it with the mean, which is 7.75. So now 5 is replaced with 7.75. Similarly, if we look at 4, so this 4 value lies in the 4th position or 4th rank, and it has mean of 6.5. So this will be replaced with 6.5. Similarly, 1 is present in the first position, so its row mean is 3. So we will replace 1 with 3. Similarly, this 3 is present here, and in the rank it is the third rank which has mean of 6. So we will replace 3 with 6. Similarly, we will do the substitution for the entire rank based data, the entire columns of the row data, but make sure that whenever you are looking for a column, so the comparison of the rank of this column will be done with the ranking of that particular column. So its rank will be seen column wise, whereas mean will be seen row wise. So that's why it is known as mean rank values. Okay, so I hope you are clear with this. So now let's move to our next slide. So there are certain problems which are associated with the quantile normalization. So now what can happen is that there are some assumptions with quantile normalization, that most of the proteome remains stable while only a small portion is expected to vary under certain stimuli. But this is not the case. In certain cases, what can happen is that your proteome might not remain stable, it might vary. So in that case, the quantile normalization can cause some problems.
So now let's discuss three scenarios. So first is the targeted changes. In this, what happens? There is small variability within the groups and small variability across the groups. So now when you are comparing, for example, three groups, then it is also possible that for those groups there is some variability among themselves, whereas there could be variability across three different groups, group 1, 2, 3. So in that case, if there is very small variability, so what you can do is you can simply ignore it. It could be a small technical variability and there are no global changes. So in that case, either you can ignore it or you can simply use the quantile normalization, but it is not necessary because their changes are very small. Whereas if we talk about the targeted changes where the large variability present within the groups, means inside the group only there is large variability and there is small variability across the groups. So what can happen is this variability within the same group can arise due to the technical variability. So there is large technical variability or due to the batch effects. So which means there are no global changes, but within that particular subset only changes are coming because of your batch effect, that how the samples were prepared. So to deal with that, you can go for the quantile normalization, and you should go for the quantile normalization in such scenarios where the changes are happening within the same group. But if the changes are global, which means that within the groups the variability is small, but across the groups the variability is large. So now one reason for this variability could be the global technical variability. That means there could be some batch effects, but you should also make sure, because these are compared between the, means across groups, that means different groups are compared. So this could also be a biologically relevant changes between those groups, because those groups might vary in their expression profiles. So that could also be one of the reason. It might not be a technical variability. So in case of technical variability, you can use the quantile normalization, but you have to be sure that this is not a biological variability, because if it is a biological variability, then you cannot use quantile normalization, as you will miss out those values which might be relevant, because this quantile normalization will not consider the biological variability and it will remove the extent or outlier values. So in that case, you should be careful while using the quantile normalization. Now the next method is two component normalization. So this is the another method in which there are two components which are normalized. So it involves normalizing the protein intensities based on the known concentration of internal standards. For example, spike in peptides and stable reference proteins that are expected to be consistently expressed across all the samples. So in this case, you are aware of the stable reference proteins and you are kind of expecting that this should be the consistent pattern, this should be the consistent expression which should be observed. And you also have the concentration of internal standards. For example, there are two spike in peptides which are observed here, and you know that the reference should be present among this region. So in this case, as you are aware, so you can use the two component normalization. This ensures that the observed differences in protein levels are more likely due to the biological variations rather than technical inconsistencies. So in this case, this is a robust method because it adjusts the values by addressing multiple sources of variability, and it also improves the reliability because now you are aware of the reference proteins, so you know that where it should be expected. And these are also referred to as the Gaussian mixture models. So you use Gaussian mixture model, and in the data you try to normalize it, you try to fit it in the appropriate distribution pattern, so that it could be more of the, it could generate more of the informative content for you. So this is all about the two component normalization. Now let's look at the missing data. So what can happen other than the batch effect is the missing data. So in certain analysis, you might not get certain values which are missed. So there are multiple reasons for missing the values. So there are different types of missing values. So firstly is the missing not at random. That means the values are not missing random. There are some biological reasons or some detection limits due to which these values are missing. So this is not the problem of the data, but this is the problem at your end or some technical limitations. So we cannot ignore these values. Whereas missing completely at random, these are the values in which the data is missing due to random technical errors or sampling variability. So when we talk about the batch effects, so those batch effects are responsible for the missing values, because there might be some variations in the sampling due to which the intensity of the data is not, the feature intensity was not that strong, so that you were not able to capture it. But this is due to the means this do not have to do anything with the data values, but this is just the technical issues or some sampling problem, the issues which are done due to the mistakes. So the experimentalist, due to his mistakes or due to some device failure or something like that, if the missing value is due to that, so that is known as missing completely at random. So we can ignore these values, means we can even simply delete them. But in case of the values such as missing not at random, then we cannot ignore them. So now in this figure it shows that this is the MS stats. So in this case, this is the general means this should be the pattern which should be captured. These all are the values, these are the intensity values which should be captured otherwise. But this red line shows the limit of quantification. That means you cannot quantify the values which are below this intensity. So this is the values which will not miss at random. This is missing not at random due to the detection limits. You were not able to detect this point. So what can happen is that if you do no imputation, that means you are not filling up this missing value. So what can happen is that you will miss out this value. So in that case you won't get any information, so you simply delete it. But in case you impute it, so you can impute it with 0. You can simply replace that value with 0, and in that case that feature is retained. But still in many cases 0 imputation could not be a better approach. But if you want to consider this feature, you don't want to miss on this value, then you can simply impute it with 0. But there are other methods also. So now let's look in the next slide. So now how you can handle this missing data. So in this figure if you see, firstly you load the data set. Now firstly what you will ask, that are there any missing values in your data set? So if there is no missing value, then you can simply proceed with other pre-processing tasks. Whereas if there are missing values, you will determine the nature of the missing data, that what is the nature of missing data. If data is there is not missing at random, so you will perform the list wise deletion. If there is missing at random, you will perform list wise deletion, perform regression or mean median imputation, or use algorithms. So these are some algorithms which you can use, machine learning or expectation maximization. And missing completely at random, then also you can perform list wise or pair wise deletion. You can perform regression, median or mean, or some imputations, and similar algorithms can be performed. But missing not at random, for them these values are not missing due to the sample issues or technical means variabilities. These are only happening because there is detection limit. So in that case you simply have to delete these values, you cannot impute them, because this is the limitation of the technical limitation. You are not able to even find that data, so that data is completely missing. That is not there is not any error, so you cannot go with the regression or mean median imputations. Now these are some kind of techniques which I have mentioned. So first is imputation, which means you replace these missing values with the estimates, such as you can either replace it with the mean, or you can replace it with the column median, or you can use certain algorithms which is k nearest neighbors. So you will observe its neighbors, you will see that what would be the expected value it should have near to its neighbors, so that you will replace, in place of missing value you will place some value. Or you can go for the deletion, which means you can simply exclude the sample or the feature with missing value using list wise or pair wise deletion. Then comes normalization. So in normalization, as we have seen, you have to adjust the data to ensure the consistency and comparability, in which you can use the techniques such as k-mean normalization, or which aligns the data by grouping the similar samples and normalizing within the clusters. So it will see that to which cluster that value falls, and in that way it will be able to replace or normalize your data, so that you do not have to deal with the missing value. It will normalize your entire data set. Then you can also use statistical models, and for these the methods are expectation maximization, in which you try to estimate the missing values based on the observed data distributions, which the data which you have observed. Now let's look into the different statistical tests which you can perform in mass spectrometry. So now after normalizing your data, after you are sure that you have the proper data which is devoid of any of the batch effects and everything, now you want to make some conclusions, you want to get into the interpretations. So for that you have to have the statistical tests, so that you would be means you should be sure that your analysis is accurate, you are reliable, and you can compare among samples. So the different tests which you can perform is t-test, ANOVA, Mann-Whitney, Kruskal-Wallis, chi-square, false discovery rate correction. So now let's discuss them one by one. So in case of t-test, you will compare the means between the different groups. So in this case, we generally go for the two groups. So we compare the mean among two groups, and if there is a considerable difference between the mean value of between the two groups, then we will say that these two groups are different. But if their mean value is same, then we cannot say that they are different from one another. So this is also one kind of test where you will compare two different conditions and you will interpret that whether they are different or not. Then comes ANOVA, which is analysis of variance. So in t-test, as I have talked, that there are only two groups, but in case of ANOVA, if you have multiple groups and you want to perform comparisons, so in that case, if you want to compare their means, you will go for ANOVA. There are two types of ANOVA, one is one-way ANOVA and one is two-way ANOVA, and this compare the differences across multiple conditions. And one more thing which I want to add about t-test and ANOVA, so which is their assumption. So these two tests are the parametric test, which means they believe that the distribution is the normal distribution or the Gaussian distribution, which means the mean lies in the center and the values are distributed symmetrically across the mean. But what if your data is non-parametric? If its distribution is not Gaussian distribution, it is not normally distributed across the mean. So in that case, you cannot go for the t-test and ANOVA. So simply go for the Mann-Whitney U-test. Mann-Whitney U-test is same as t-test, but for the non-parametric test, it will also compare two independent groups when the data is not normally distributed. Similarly, Kruskal-Wallis test, so Mann-Whitney is used for the comparison among two independent groups, whereas if you have more than two independent groups, so you will go for Kruskal-Wallis, which is the non-parametric test to compare three or more independent groups. Now if you have the categorical variables and you want to assess the associations, so you will go for the chi-square test, which is used for the categorical variables. That means the values which are not in the numbers but in the categories, like yes, no, affected, not affected. So in that way, so this, for that you will use chi-square test. Similarly, false discovery rate correction is also a test in which you will adjust for multiple comparisons to control the false positives. So you kind of adjust, you correct the p-values. So we will see the false discovery rate correction in the multiple hypothesis testing. So what we do is we try to correct it. After performing the statistical tests, there could be some issues which might remain. There should be some false positive values which you might observe. So you have to deal with those values, for which we go for the multiple hypothesis testing, in which you try to control the rate of false discoveries and ensure that the statistical inference which you have drawn is reliable when you are analyzing large data sets with many comparisons. So such methods are Bonferroni correction, which tries to control the type 1 error rate. So what it does is it adjusts the p-values by dividing it with the number of tests which have been performed. Then the use case, that when it is used, so it is a simple and conservative and suitable for small number of tests. Then comes false discovery rate correction, which controls the expected proportion of false positives among the rejected hypothesis. It's also adjusting the p-values using the methods like Benjamini-Hochberg. So we will discuss that what is this Benjamini-Hochberg method. So these all methods, if you see, they are trying to deal with the type 1 errors or the false discovery rates. So one is Horn-Bonferroni method, which control type 1 error rate with less conservatism. It sequentially adjusts the p-values starting from the smallest. So p-values are like the confidence values, and it is mentioned that for any statistical test to be significant, its p-value should be less than 0.05. And if it is higher than 0.05, then that means we cannot accept that hypothesis. So p-value should be lowest to be significant. So that's why in this case we are trying to sequentially adjust that value starting from the smallest. Then comes Benjamini-Hochberg procedure, in which it controls the FDR by adjusting p-values based on their ranks. So it ranks p-values and adjusts them to control the FDR. So it will see that if still we are getting false positives, so it will make sure that the p-values should be strict. If p-values will not be strict, then many false positives could be captured. So what is happening in all these methods, we are trying to restrict our methods to capture the results, to make sure that there are no type 1 errors, no false discovery rate, no false positives being detected. So this Benjamini-Hochberg procedure and these procedures, they are trying to deal with the p-values, which are the confidence values. So they should be smallest to be to accept anything as statistically significant. So if p-value is means if certain p value is still giving you the false positive, so you try to reduce it further, so that till the point where the false positives are not present in your sample. So this is all about the different methods which are generally used to deal with the false discovery rate as well as type 1 error rates. Now let's look at the MetaboAnalyst. So this is the hands-on the MetaboAnalyst. So a MetaboAnalyst is a web-based platform which is generally used for the statistical analysis, visualization and interpretation of the metabolomic data, and it offers tools for the data processing, statistical analysis and functional interpretation. So you simply have to upload your file and this will perform all the analysis which you want to do. This is the home page of MetaboAnalyst. You can go through all these options. There are tutorials which are provided by them, so you can study them. Now let's see at the example. Now you can click on this link. It will redirect you to the particular server. Now what you can do here is, if you want to perform the analysis, first step is to upload your file. So firstly you have to select the data type. So there are three options for the data type. One is concentration, second is spectral bins, and third is peak intensities. So it depends that what data you have. For example, I have a metabolomic data where I have multiple proteins. I have done its quantification and I have concentration of each and every protein. So in that case I will select concentration. Whereas if I have mass spectra data and I have the spectral bins, I have different spectras, so I want to compare them. So in that case I have to select spectral bins. Whereas if I have intensity values, so I will select peak intensities. So it depends what kind of data is present in your file. Then comes format. So in format, it depends what is the format of your file, how the data is arranged. So in this case my data was arranged in the columns. So samples were arranged in the column wise and they were unpaired. So it is also possible that if my samples are arranged row wise, so in that case I have to select the format, that how my data is arranged, so that it can distinguish among different samples. And now I have to upload my data file. So in this case make sure that the data file is in .csv format, because it only accepts .txt and .csv. So even if you have .xls format, which is the Excel file format, so just go to the MS Excel and save as .csv. So in file format you can select for .csv or .txt, and then you will be able to access this file and submit it. Now after submitting, the result would appear like this. This is the initial initial result, in which it will tell you that they have checked the data content, it has been passed, samples are in columns and features are in rows, and uploaded file is in .csv format. It contains 15 samples and 2548 compounds, data matrix. So these all things will be present here. So it will give you the whole crux of the file which you have given. Samples are not paired, 2 groups were detected. So these all things will be given. Along with this, it will also tell you that whether there are some missing values or not. So they are saying that 0% missing values were detected. That means 33 features with a constant or single value across samples are found and deleted. So in certain cases, if there are any missing values, or there are some constant or single values, so they have been deleted. And as now there are no missing values, so by default missing values will be replaced by 1/5 of the minimum positive values of their corresponding variables. This is by default method, but in case you want to change this method, then you can simply click on missing values. Now in this case they are saying there are no missing values, so this missing value is not highlighted, it's blur, it's light, it's faded. It means you will not be able to click on this. So in such scenarios you can simply click on the missing value present on the left side panel. So if you click on missing value, you will be taken to this particular pop-up window where you can select that how you want to address the missing values. So you can remove the features with greater than 50% missing values. So if there could be multiple features which could be present. So if in certain sample, if multiple features are missing, so you can simply remove those features if they have more than 50% missing values, and then estimate the remaining missing values. So in case I don't want to remove values, or in case the missing value percentage is lesser, so in that case how I want to deal with it. I want to replace it with log odds, that is 1/5 of the minimum value of the each variable, or I want to exclude variables with missing values, or I want to replace it by the column, or estimate the missing value. So I can also use KNN, or even I can replace it with the mean value, median value. So all those things you can select here, and then you can click on process, Now it will take you to data filtering step, where everything is by default present. If you want to change it, it depends on you, but I would suggest that go with the default, because it depends. When you are aware of these, then you can select options, but if you are not aware, then go with the defaults. For example, in abundance filter they have selected mean intensity values. So in case of mean intensity values, why people go for median intensity? Because mean is very much influenced by the outliers. So if in your data, if there is some outlier value, then mean will be shifted towards the outlier, whereas median is not shifted that much towards the outliers. Or you can say if there are difference in the values, so if one is very low, one is very large, so this kind of data is present, so in that cases mean can get influenced, whereas median won't get influenced. But uh, it's up to you what you want to select, so I am not changing anything, and we will simply click the proceed. Now next step is the normalization overview. So in this case there are different sample normalization methods. You can select any which you want, that how you want to normalize. So in this case by default none is selected. Then data transformation, so you can also transform your data. For example, if your values are very large and you cannot see them properly in your table, means in the graph, so simply you can go for the log transformation, so that the range can be adjusted. Or you can go for the square root transformation, cube root transformation, depends. So now the default is none. Similarly you can scale your data, so it all depends on you, but in this case we'll keep it default. And now we can, if you want to change, you can simply click on proceed. If you want to normalize, click on normalize, and then if in this case we don't want anything, so simply we'll do view results, and the results will appear like this. So as you can compare among normal, means before normalization and after normalization, so there are not much effects. I think there is nothing is changing, so which means that our data set was not having any such issues with the sample preparation or any other issues. It was already, means it was not having any such variations. That's why after normalization also our sample appears to be similar. So this could be one of the reason. And if you want to further analyze, then you can go through this panel. You can select multiple options. So I have selected statistics. So there are different statistical tests which you can perform. You can look for the fold change analysis, that because it has multiple proteins and multiple samples, and now they are comparing that which of the proteins are changing among them. So for fold change, you can see that how much change has happened among different groups, among different samples. Or you can also go for the volcano plot. So in this I have selected volcano plot. Now what it will show you, this volcano plot, it combines the result from fold change analysis and t-test into one single graph. So now it has combined, it is giving me log p value also, which is the confidence score which is taken from the t-test, in which the means have been compared, and log fold change. So both of them are represented in the similar plot by a volcano plot. So these red dots represents the significantly upregulated proteins, whereas the blue dots represents the significantly downregulated proteins, which are 243 and upregulated are 58. If you pass your cursor among any of these dots, it will also give you the IDs, uniprot IDs of that particular proteins. So in this way you can simply analyze your data, you can visualize your data effectively. You can see that what is the difference among two different samples, are your results significant or not, because p value is also calculated. So p value is also giving you the idea that whether the variation which you observe, either they are significant or just random. So everything you can do with the metabo analyst. So this is all about metabo analyst. Now we will quickly practice few MCQs which will help you to solve your assignments. So the first question is that there is a Raghav who has performed a mass spectrometry experiment for the first time for optimization, and based on his observations from this experiment he modified his further experiments. So these are the first initial experiments which he has run, and now he want to modify that which one is better, so he'll accordingly proceed with that particular method. So following figures shows the three different ms1 chromatogram profile for experiment 1, 2 and 3. So this is the chromatogram. So in this case the y-axis will be the time in nanoseconds, and x-axis is time in nanoseconds, and y-axis is the relative abundance of particular peptide in that particular time point. So assume that he loaded equal amount of samples for each experiment. Now you have to answer the following questions from 1 to 4 based on the following chromatograms. So if you see clearly, this is chromatogram 1, in which the time is ranging from 0 to 44, whereas the peaks were observed nearly around 15 to near about 35, 36. Whereas in chromatogram 2, the elution time was nearly 120 and the peaks were observed from 20 till 110 to 120. So this is taking longer time, multiple peaks. This is taking shorter time. That means elution is quite fast, whereas it is taking some time to elute. Whereas in chromatogram 3, if you see, so its time is from 0 to 180 and the peaks were also from nearly from approximately 15 to 160 and the larger peak is obtained at 173. So this is taking even a longer time and the peaks are also sharp, that the abundance is high. That means these are not short peaks, so which means that this is also significant peaks. Now let's see the questions. So first question is that Raghav used the data obtained from these experiments for protein identification, which was done by MaxQuant search engine. Which of the following statements are most likely to be correct for result obtained from 3 experiments? So the first is there is no difference in the protein coverage, only a small difference in protein coverage, and a considerable difference in protein coverage, or insufficient data to make any conclusion. So let's look at this. So do you think there is any difference in the protein coverage? Okay, so yeah, we can see that elution time is quite variable among both, and the intensity values or abundance is also variable among all these, so which suggests that there is the difference in the protein coverage among these. So the correct answer will be option C. What you have to do, the chromatograms from these experiments, they show variation in the peak distribution, intensities and overall profile, which suggests that there is a difference in the underlying data, and these differences in peak profiles across these experiments are significant, because I have shown you that in case of this, these abundances was also, this value was also higher, 93.62. This is not insignificant, this is quite significant because abundance is high. So similarly means we should be aware that their abundance is high, so we should be take that into consideration. So that's why we can say there is a considerable difference in the protein coverage. Now let's look at the second question, which says you have to choose the correct order of protein coverage obtained from 3 experiments, and these are the options. So which has the highest coverage? So as we have also discussed that experiment 3 was having the higher coverage because it was having the greater retention time and peaks were also strong. Then comes experiment 2 and then experiment 1. As you can see that these two have the higher peak values and greater coverage as compared to experiment 1. So if you have to order them, so it will be ordered like experiment 3, then experiment 2 and then experiment 1. So experiment 3 has highest protein coverage due to its broad distribution and significant peak intensity, followed by experiment 2 with slightly less comprehensive coverage, and experiment 1 with least coverage due to its condensed peak distribution, because it is eluting quite fast. So that's why the peak are very condensed, they are very close to one another. Now let's look at the third question. So which of the following gradient in liquid chromatography could be used by Raghav to obtain the best chromatogram? So those 3 chromatograms we have seen. So what should be the gradient which he should use? So the given graph is A, B, C and D, and you have to select one among them. So now before answering it, let's look at the what does it mean. So the correct answer I'll tell you, it's D, because you can see there is a stable, constantly you are rising, and then there is stable rise in the AB concentration, A to B concentration. So I'll just give you the idea that how it works, so that after that you will be able to understand that how the option D is correct. So what happen is in the gradient, so a flow rate is measured in nanoliter per minute, which is nano LC in nano liquid chromatography. So solvent A and B, so solvent A is water, solvent B is acetonitrile. So as we know, the liquid chromatography which is used for mass spectrometry is reverse phase liquid chromatography, in which what we do, we separate the peptides on the basis of the hydrophobicity. So hydrophobic are the peptides or are the compounds which are not soluble in water. They are hydrophobic, they are water hating. So hydrophobic compounds or hydrophobic residue containing peptides, they will elute slowly if the solvent concentration is high, if water is greater in the mobile phase. So in that case hydrophobic will elute slowly, whereas if acetonitrile, which is hydrophobic in nature, so if that is present in the mobile phase, so my uh, peptide present in the sample, they will elute faster, which are hydrophobic in nature if acetonitrile concentration is high. So in reverse phase liquid chromatography, we increase in hydrophobicity to elute out the hydrophobic peptides. We gradually increase the hydrophobicity such that firstly the less hydrophobic will elute and then more hydrophobic will elute. So in practice you will start with the high proportion of solvent A, that is water, such that more and more hydrophilic peptides will elute out. Then a point will come where when you will be left with the hydrophobic compounds. Now what you will do, you will gradually increase the proportion of solvent B, which is acetonitrile, in a mobile phase gradient. Now what it will do, if you will increase it gradually, now it is gradually increasing the concentration. Now what it will do, it will slowly and slowly try to remove the hydrophobic peptides which are present. If you will increase it suddenly, then what it will do, it will completely elute out all those hydrophobic peptides at the same time. But your aim is to increase your resolution, properly distinguish among different intensity of hydrophobic peptides. So for that you should increase it steadily, and it should not be like a line, straight line, it should be like increasing steadily. So that's why if this is the steady increase in the concentration of acetonitrile in the mobile phase, which will help us to compare among the hydrophobic peptides clearly. Now let's move to the question again. In A we can see there is a zigzag. So if there will be a zigzag concentration variation among the AB grade, means AB solution, so it will confuse us, because at one level it will elute out hydrophilic, then at other hydrophobic, so we won't be able to distinguish properly among both the peptides. Whereas if we use the B, so in this case although it's increasing steadily, but you see the concentration is constant, it's not that much increasing, it's constant to a longer time period. So in this scenario we won't be able to properly move out the hydrophobic compounds, because they will move slowly, and then you are suddenly rising it. So what will happen, till here only fewer peptides which are lesser hydrophobic they will move, and then suddenly all the other hydrophobic will move out suddenly. So this will also not help you. Similarly in C they are decreasing its concentration, so this will not help you to observe the hydrophobic properly, hydrophobic peptides. Whereas D, it means it appears to be feasible, they're slowly increasing the concentration, slowly moving out the hydrophobic compounds, and ultimately increasing it to a greater extent, so that all the hydrophobic compounds which are left, they would elute out, or the peptides which are left, they are elute out. So that's why option D will be correct. All will not give the same or equal result. So I hope you are clear with this. Now let's move to the next question. So according to above conclusion, which experiment would you suggest? Because we have started that Raghav has done three experiments. This was his initial set of experiments. Now he want to proceed with the experiments of which one he should do. So after all the analysis, we have seen that experiment 3 was showing greater coverage in the chromatogram, so that was better, so he can proceed with it. That's why the correct option is answer 3. Now let's look at the next question, which means which type of missing value is non-ignorable? Which of the missing value we cannot ignore? Option A, missing at random. Option B, missing not at random. Option C, missing completely at random. And option D, both A and B. So as we have discussed that the values which are missing at random, they can be ignored because they are randomly missing. Whereas if values are missing not at random, then we have to carefully see that what could be the possible reasons. There could be biological reasons behind it, there could be technical limitations. So these values need to be addressed and these cannot be ignored. So I have given an example, means how you can remember these things. So for example, missing completely at random, that means dog eats any homework. Somewhere somebody have done some homework and that has been eaten by dog, we don't know any of the homework, it's so much random. Then comes dog eats student's homework. So some student is coming to class and dog has eaten his homework, so that means it is missing at random because the student is not decided, but that student's homework is eaten, that was the homework of a student. So student is still a little bit not complete, and it's not completely random but still at random. In class multiple students can be there. Then comes dog eats bad homework, which means that this is not at random because it has selected bad homework. dog has decided which homework is bad and then has written specifically the bad homework, so which means that is not missing at random. So that's why in such scenarios there could be possible reasons while some values are missing, so we need to handle them properly. We need to perform either their pairwise deletions or we need to deal with them properly. We cannot ignore them. Now let's look at the next question. Which of the following will help you to remove the technical artifacts? So technical artifacts, as we have discussed about the batch effects. So first is log transformation. Log transformation is not done to remove the technical artifacts. Then data scaling, this is also not done for technical artifacts. Batch correction, yes, it is done for the removing of technical artifacts. And data munging, this is also not done for technical artifacts. So correct answer is batch correction, because these technical artifacts are generated due to this improper sample handling, or there are some issues due to some machines. So these all can be corrected with the batch correction. Yeah, so this is all about your session. I hope you enjoyed, and let me know if you have any doubts in the comments. Thank you so much.